see in Headco Neuroscience building, but his PhD was in the computer science, while I was at the same time in neuroscience. So we overlapped, and it was the heydays of the Michael Arbib's lab, and it was something. And we can talk about this if you're interested. That was a pretty creative environment at the time, and very dynamic. Then, sorry, Peter.
Indeed, yeah.
Indeed. Then Peter had the kind of opposite track to Peter and I were the opposite track. He was an American and he moved to France, and he worked at the CNRS, and now he became a director in the CNRS, and he's now he spent a lot of time in Dijon in the Jean lab, and then he now moved in Lyon in the Jean lab, and now he moved to Dijon. And he's working with actually one of my collaborators, Haris Papaxantis, who came and gave a talk, I don't know if you remember, in imagery. Peter is a very creative researcher. He has many interests. He's interested in motor control, but also in cognition and in robotics. And his research is kind of, an umbrella on these different topics. And so today he thought he had a topic that may interest us in rehabilitation. And so this is how we came up about this idea of him giving a talk here today. Peter, thank you very much.
Great. Yeah, thanks. Thanks, Nicolas. And indeed, yeah, it's that was quite an interesting environment. We had Michael Arbib created a really rich environment that continues to reverberate today. So today I'm going to share with you some results and new protocols, preliminary results in the area of time and space in motor sequence learning and possible uses in re-education. So probably I don't have to tell you that motor behavior is organized in space, but also in time. And so you can think of things like rhythm in piano or drumming. So the temporal structure of motor control is of equal importance as the amplitude of the movements themselves. And both of these dimensions can be manipulated and can be used to enhance learning. And so one fundamental research question is, do they rely on dissociable neural processes? Is time processed in one way and space in another, or a common process, or both? So this is something we can look at, and that may be of interest in re-education or rehabilitation. And so we have kind of a novel approach where we're using a new paradigm that uses robotics and state-of-the-art motion capture to evaluate this kind of the effects of space and time in motor learning. And we're also looking at EEG correlates of this. And so my objective today is to share this with you, and I hope we can have some discussion. I'd be really happy to get feedback about potential applications in things like stroke rehabilitation. And so just a warning, these are preliminary results, but they are some results that we can talk about. So back in even before Nicolas and I were, like when we were PhD students, the Nissen and Bulmer studied and they invented what's now called the serial reaction time task. So this is a classic task used for assessing motor learning. And the way they did it back in those days was, you see on the left here, people have their fingers over a keypad, which can also be a computer keyboard, and letters on the keyboard are associated with diodes that can light up. And so basically you show a diode, the person responds with the key that's close to it. That triggers the next stimulus, the next light that lights up. And so basically it's a key tapping sequencing task. And so what they did is they studied sequence learning in this context. So they made up a 10-element sequence where A, B, C, and D are mapped onto these four, not using the thumb, mapped onto these four lights and four key presses. And then they presented these in continuously repeating sequences in a block with 10 repetitions of this sequence per block. So that makes 100 trials, which takes about 1.5 minutes or so for the people to do. And then they have 8 blocks of 100 trials. And so what we see on the right here is this classic serial reaction time learning curve, where you see for trials that were organized according to this sequence that repeated, you see that the reaction times, so they're measuring reaction times to each key press, go from about 340 milliseconds down to about 180 milliseconds. So a nice, robust reduction in reaction time. Whereas with control subjects who were seeing just random sequences of these button presses, they had a small motor learning that gave them a slight advantage over time, but not this big sequence learning effect. So this, I don't know if you use this task or if you're familiar with it. It was hugely used in experimental psychology. There are all kinds of manipulations that you can do related to the complexity of the sequence. So one thing that we tried to study was looking at the spatial and the temporal aspect. So in 1998, we were using in the lab in Lyon touch screens for doing sensory motor behavior. So we made a version of the serial reaction time task using a touch screen. So it's basically the same idea, but instead of responding on a keyboard, you point to targets that appear on the screen. And so we had the same kind of setup as Nissen and Bulmer, 6 blocks of 80 trials. Each block was made up of 8 times a 10 trial sequence. So, and I think this sequence was even taken from their work, but we made a modification in order to study temporal structure. So in the training blocks, in our experiment, we exposed the people to 6 blocks of sequences using this structure, but where targets A and D are preceded by a response to stimulus interval of 0.2 seconds, and B and C are preceded by a response to stimulus interval of 1 second. And so when you see people doing the sequence pointing task, this structure of response to stimulus intervals makes kind of even a visually visible rhythm to the sequence. So it's like da 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 da. And so so we see then we have we have 6 blocks where they're learning the sequence. Then in the 7th block, we swap the temporal structure. So now A and D are preceded by the short delay and B and C preceded by the long delay. And then in block 8, we go back to the training sequence. And then in block 9, we change everything. We change the serial order and the temporal structure. And so what you see here is the reaction times for people in the first block, 2nd, 3rd, 4th, 5th, 6th. So we see this nice classic learning. And then in the 7th block, we see the classic serial reaction time increase in reaction time, when normally in standard serial reaction time tasks, the test block is just a random sequence. So and then you see the difference between learning advantage, so the reduced reaction times because you know the sequence, versus reaction times when you don't know the sequence. Here we see this increase in reaction time to a change, but it's a subtle change. It's just a change of the temporal structure, but not the spatial locations in the order of the sequence elements. So that's an interesting result that says that if you can keep the serial order and the spatial order identical, but if you change the temporal structure, that will perturb people and give these increased reaction times. Then we go back to the normal sequence, and then finally a random sequence where the reaction times go way up. So that let us make some conclusions about the idea that the sequence is learned in kind of a combined spatial and rhythmic or temporal manner. And so, but there's some limitations and some questions about generalization. So a limitation in this kind of experiment is that you only get response times. You don't know what happened in the trajectory, for example, when the person was pointing from one location to another. When you, for example, when you change the temporal structure and you increase response times, will you also modify parameters of the trajectory? With this serial reaction time methodology, we can't measure those things. So Nissen and Bulmer started with buttons on a keyboard. We moved to pointing movements on a touch screen, but now we can ask the question, will this generalize to more extended movements? And so can this method be exploited for a broader range of movements? And so here's where we move into kind of the modern world. So we have a new behavioral paradigm that exploits state-of-the-art technology for teaching and evaluating learning. So what we see here is in our laboratory in Dijon, a big room for motion capture analysis. So these are Vicon cameras oriented around a humanoid robot. And so this is so what we can do is use the robot for generating stimuli to train people and then record the movement of the robot and the movement of the people in these sequence learning tasks that I was showing you. And so here's a video. I don't know if you can see this moving. Do you see something moving? So what you see here is the robot is now performing the same kind of sequence that we were using in our earlier experiments. So we have 4 spatial locations in the peripersonal space, in the shared space between the robot and the human. You can think of them as being labeled A, B, C, D. And so these same sequences that we were manipulating in the serial reaction time task, we can now use in this human robot interaction task to study spatial and temporal properties of sequence learning. So in this new modified protocol for face-to-face imitation, again, you see the person is sitting comfortably in a shared space with the robot. The robot is making these movement sequences and the person is following them. So our hypothesis is that humans will implicitly learn a combined spatial and temporal movement sequence from robot demonstration. And that if we change this temporal structure, but leave the spatial order intact, we'll see a disruption of the learned behavior, which would be evidence that the people learned the temporal structure as well as the spatial structure. So the method is so now instead of measuring reaction times to somebody touching a keyboard or touching a touch screen, we're going to use 3D motion capture, and then we can measure things like the velocity peaks or acceleration peaks or just position of the hand of the robot and the person. So we evaluate the performance by the lag between the human and the robot. So this is just an example of a trace where in red, pink is the robot movement and blue is the human. And so you see in this time period, the human is is behind the robot. So there's a lag. So that lag is the performance measure that we can use. And so we're going to do the same kind of repeated block structure. So we have 9 blocks of sequential pointing movements. Each block is made of 5 repetitions of a 16 element sequence to make 80 movements. So each block takes about 1.5 minutes to do. So there are 9 blocks, and we've got about a 30-second pause in between blocks. So and so we have the same kind of procedure where there are several blocks of trials that use the same sequence for learning. So in our case, that's blocks 1 through 4. So there we expect to see some learning. Then in block 5, we do a test. And the test is we keep the same spatial sequence, but we change the temporal structure. Then we have 2 more blocks 6 and 7 that use the same training sequence. Then block 8 tests by modifying the spatial order, so the order of the elements in the sequence, but keeping the same temporal structure or same rhythm. And you can see the kind of a schematic. So the training sequence, when there are 2 blocks, when there are 2 dashes, that means there's a 500 millisecond delay. And when there's 1 dash, that means no delay. So the training sequence and the test sequence are the same in terms of the order of elements, but they have different temporal structure. And then the second test sequence in block 8 is just the reverse of the training sequence. So what we're gonna, and this when we made this protocol, it was almost to kind of verify that our robot interaction and Vicon system worked, because we could pretty strongly predict the results that we would see. We should see a learning curve for the first 4 blocks. Then in block 5, an increase in the lags. Block 6 and 7 should return to the lower lags based on the learning. Then block 8 should increase, and block 9 go back down. Please feel free to interrupt if you have questions. And so here, what we see, so what we see is lag increases when the spatial structure is the same and temporal structure is modified in block 5, and also when the spatial structure is modified and the temporal structure is kept the same. So that's what we see here in this graph. So this is, here we're calculating the lag between the XYZ position of the robot hand and the human hand in the 9 blocks. So blocks 1 through 4, training. So we expect to see the lag go down. Then in block 5, we manipulate the rhythmic or temporal structure, but keep the sequential structure the same. The lag increases. Block 6 and 7, we go back to the original training sequence, so the person recovers. In block 8, we change the serial order. So then we see a big increase in lag. And then the lag returns to low. So there's this progressive learning curve, and then superimposed on that are the 2 blocks where we manipulated the sequence that had been learned. And here we show the combined trajectory for 1 block. With red is the robot movement and blue is the human movement. So already one good thing is that there's very little variability in the robot movement. So the robot is fairly fairly low variability. It's reproducing its movements fairly well. Whereas in the human, we see a fair amount of wagging around, which we'll talk about. Is that dispersion something that can be analyzed as a behavioral factor, indicating more or less success in following a sequence? So just to do the statistics on these results. So we had 9 subjects. 5 had the time modified in blocks 5 and the space modified in blocks 8, and then 4 had the reverse. We ended up combining them because there were kind of equal deficits produced by these manipulations. So there's a significant effect for block, and then post hoc tests show that in the 2 blocks where we modified things, in the 2 blocks where we modified things, there's a significant increase in the lags for the modified block versus its 2 neighbors that were unmodified. So block 5 compared to block 4 and 6, and block 8 compared to block 4 and 7 and 9. So that was that let us conclude that both spatial and temporal structure were learned, and if you modify one independently of the other, you produce these increase in behavioral lags. So at this point, we're really not completely exploiting the richness of the data that we have, because we're only looking at these lag times. So, and this is something that maybe we can talk about in the discussion. I don't know if you go down to this level of motion analysis in the kind of work that you do to look at the, but so when you look at these trajectories, this is just for one short period. So the robot is ahead of the human making his movement. The human is several time steps behind, catches up, there's a pause. So already you can see that this data is fairly rich. You can analyze things like smoothness and jerk or deviations in trajectory. So these are kind of open issues that we're still looking at. But in terms of dispersion of motion in the trajectory, so here again, I'm showing you on the left for a different subject, the reduction in lags. And then I want you to focus on the block 4 and 7, because those are the blocks where the lags were lowest. And then the following block 5 and 8, where we modified the sequence and where we saw an increase in the lag. So I hope by now this format is familiar to you. So if we look at and this is a very gross way of looking at the data, but just looking at the data grossier from all, just superimposing the trajectories for the whole block. In block 4, we see, if we compare block 4 and block 5, when I look at that, I see more dispersion in block 5. So, and that would be consistent with the idea that, well, yeah, because in block 5, you've perturbed the sequence, the person's lag times increased, so clearly they're kind of struggling and they don't know where to go. The learning that they've done before doesn't apply as well. And so that would explain or predict that you could see increased spatial dispersion. And then similarly, but maybe a little bit less if we compare block 7, where we're back to the good sequence that the subject has learned, and then block 8, where we change the order of the movements. Looking at this, it's like looking for galaxies in your telescope in your backyard. If you really look, you can maybe see some increased dispersion here, but this is something to be further analyzed, and I'd be interested to have your opinions on whether that's something that would be
Peter, could I ask a question, clarification? How do you measure lag?
We're using a correlation function in MATLAB that just compares that tries to align the signals based on either velocity. So we use either position or velocity, and then it's basically aligning the peaks and troughs in those signals.
So it's sort of it's not, you're not taking at a single point in time, you're sort of more sort of, it's sort of an average lag, if you will.
That's correct, yeah.
Because it seems to me that, and I mean, I think you're sort of leading in this direction a little bit, but I mean, looking at some of these trajectories, well, let me say, I mean, either the subject could delay the onset of their response, or I'm talking particularly now when they have when there's been a change in the temporal or spatial structure of the pattern, they could either delay sort of a classic reaction time change, or they could initiate the response but make corrections during the response. And it seems to me, I'm seeing evidence of that in some of your trajectories, that they're actually, that it's an ongoing, it's a correction that's happening in mid-flight in some cases. And I just wonder if that's something you're looking at.
That's a great question, and that's something that we probably should look at. So and kind of the simple response is, when we were first looking at this data and saying, oh, well, how should we analyze this? So I was discussing with my colleagues in Dijon who do this kind of thing. And we were thinking about, well, maybe we should segment the movements and individually for each movement, find the, so you can see in these places when there's kind of a stop before, so you move and then you kind of stop and then you move again and stop. So we could do some kind of automatic segmentation and identify for the robot and the human exactly for each of the 80 movements in each sequence, the onset and offset, and then do our measures in a much more fine way, movement by movement. And even like you said, go even closer and say, look at, okay, we know from the sequences which movements from one target to another are going to be the hardest for the subject to predict, because here's where the delay is changed. And so we could analyze those more specifically and look at these trajectory deviations that you're talking about. And so
yeah, this is an interesting one, Peter, because if you look at this, I apologize for doing this, but if you look at this, this part of the human trajectory here, it actually reminds me of what we saw in a number of studies that I participated in, where we were looking at discrete movements, not sequences like this, but in which the subject's kind of cheating. The subject is kind of sort of moving along the trajectory, and sort of getting started in that trajectory and kind of waiting to be sure that that's the direction that he or she should move in. And one often sees that we did a series of studies. I was with Claude Guez at the time. We did a series of studies using what we call a response specification paradigm, which allows one to look at what happens when subjects aren't completely sure, but are forced to respond before they're completely sure of how they're supposed to move.
Okay.
And we would often see these kind of little cheating movements. Now, I'm not sure that that's what this is. It could be just a drift, but I think it would be interesting to determine whether subjects are making those kind of early guesses but hedging a little bit before they fully commit to it.
Yeah, no, that's a great, that's exactly the kind of comment I was hoping to hear. And so I'm really happy to know that even if, that this looks like a potential evidence for behavior that is known when people are under uncertainty. So they, like you said, they may kind of start moving, but there's something not quite right, and then once it gets right, they can keep going.
Yeah, the key finding that we found, back then, and was that the determination of what we call response specification of the direction or magnitude that the response was supposed to be is not something that happened all at once, but it happens gradually, and subjects actually, it's almost a kind of probabilistic kind of function that as time is progressing, the degree of certainty on which direction or which magnitude they're supposed to produce increases. What we were able to do is sort of force them to, and you're kind of doing that a little bit here. You're kind of sort of forcing subjects to make responses even when they may not feel fully certain about what they're supposed to do.
Yeah, by having these blocks where we're changing the the structure that they become familiar with, then we force them into this unfamiliar zone where they're going to make this kind of behavior. Okay, good.
I
Yeah.
think this is a fascinating paradigm and has a lot of richness that could be exploited.
Okay, that's what I wanted to hear. That's what I think also, but I'm happy to hear that from somebody who knows more than I do about this kind of thing.
Jim and Peter, two comments question here. Jim, when you said time, you mean preparation time, correct? You don't mean time in the learning time. But it's maybe both.
I mean
right?
time during the actual development of the response specification. I mean very short period. I don't mean time over trials. I mean time within a single trial.
But the time of a trial can have an effect actually in the changing of this preparation time, maybe.
Absolutely, absolutely.
And Peter, here, this is a movement early on during training, or this is?
Yeah, I think this is in, it says block 1, so I think that's early on.
Okay.
Yeah, but it would be, like you're saying, there's rich data here, and so we can go look at these things. We need to develop some tools for automating the, but like Jim was saying, it could be that at this point, the human is kind of hesitating. So he's starting to move. He thinks he knows where to go. Maybe the robot has a delay, and finally, oh, okay, yeah, then he starts going. So that can be confirmed. But so thanks for those comments. Yeah, and so yes, and so dispersion or these temporal hesitation kind of things we can look at. So and in the quest so that would be, those could be biomarkers. What we're talking about could be a biomarker of learning, and eventually we can artificially create perturbations by modifying these sequences, so we can try to find biomarkers for perturbed behavior. But maybe there's also the goal to try to find biomarkers for people who have perturbations, for example, in the temporal organization of their movement, to see if they could, if they would be related to these markers that we can create artificially. So we've got two domains, possible domains of biomarkers. One is behavior that we were just talking about, and the other is neurophysiology. So we could use EEG or NIRS or some other kind of recording in this paradigm, in this protocol. And so we've got colleagues in Dijon and Paris, a company called BioSerenity that makes that is interested kind of in telemedicine. And so they have a disposable EEG electrode system that we recorded a subject with in this experiment, with the goal of trying to see, are there neurophysiological markers for the kind of learning that we're seeing. And so this was sort of uncharted territory for us. And so looking in the literature, one thing that comes up is this notion of event-related synchronization and desynchronization. Event-related synchronization and desynchronization, with the idea that in many kinds of motor control, when you execute a movement, you have desynchronization, so reduced power in certain bandwidths when you look at the EEG signal. So, and let me just say that in this kind of protocol, if you say, oh, we're going to record EEG data, what kind of analysis would you recommend? Well, we quickly realized this is a difficult domain if we want to try doing ERPs, because as you know, in an ERP study, it's nice to have a clear TAC, a clear signal upon which you can align your EEG data. And in this case, since we've got continuous movement, it's kind of not the right paradigm for ERP. And on the other hand, for doing time frequency analyses, this is more suitable. And so this is the kind of thing that we can do. So in this study by by Hervold. They show subjects doing rhythmic movement, and when they continue, they see a continued low power in this this frequency bandwidth. And then when they have the subject stop, so they're making kind of rhythmic pointing movements back and forth. And when they stop, then you see an increase in power. So we thought that this could be a signature for a movement that we might see in our experiment. And so in fact, this is looking at one frontal electrode, and this is from one subject, data that was recorded. So these are really preliminary data, but it looks like we have the 9 blocks here of movement and this nice bands of desynchronization during the time when the subject is moving, and then indicated by blue, reduction in power, and then the yellow-green increase in power during the intervals of rest in between the blocks. And so if we look in a little more detail, kind of zoom in on that, then we see these, so during this period of about 80 seconds, we see this desynchronization during the movement sequence, then kind of an activation during the rest period and desynchronization. So it looks like this event-related desynchronization is produced in our protocol. And then we can ask the question, is it possible that some property of this ERD signal could be related to the behavioral phenomena that we observe? And so again, this is a very early preliminary data exploration. But if we look at the power in the low power ranges from 2 to 6.5 hertz, and so this is like a raw signal of the power at 4 hertz for block, and we see the desynchronization in block 1. So these downward pointing peaks are the desynchronization at time points in each one of the 9 blocks. And so what we see is in block 5 and block 8, where we perturbed things, it looks like the desynchronization is going even farther than in the 2 preceding normal blocks. And so kind of just a simple, and it's not a reliable for publication, but just sort of a back of the napkin analysis of the power for the 2 blocks that precede the sequence that changes, either for the changing the serial order or the temporal structure. We see it looks like an effect that the desynchronization is increased, as if, looking for sort of a phenomenological explanation. It may be that people are having to increasingly allocate resources during these blocks where we've changed something because it's increasingly difficult. And so that might explain the increased desynchronization. But again, these are really early results, so kind of speculative at this point. So I've tried to show you this face-to-face imitation and make the initial conclusion that face-to-face imitation with a robot is a promising method for studying human sensory motor sequence learning. There's a variety of motion parameters that can be analyzed, like we were already discussing, and probably a variety of EEG parameters that can also be analyzed. Potential biomarkers for learning and perturbation in learning can be identified. And it just strikes me now that, so this is when we're looking in the literature for related experiments, we found a few experiments that had such extended data collection in a single trial, if you will. So we're collecting data in, during a period of almost 20 minutes, and trying to do these analyses, even in the shortest granularity of blocks of 80 trials. So that's kind of an unusual thing to do. And even my colleagues in Dijon were saying, yeah, normally we look at single movement, single pointing movements. So one trial is one movement, and that way things are very cut and very segmented. And so what we're doing here is very interesting, but it's different. And it just reminds me of a new trend in cognitive neuroscience of language. People used to study syllables and then words, and they do ERP studies when people listen to words and maybe sentences. But now people are recording in the scanner brains of subjects who are listening to half-hour segments of a movie, like or a TV series, Sherlock, or the regular movies. So and this requires new kinds of analyses, but maybe there's a trend towards more ecological kind of experiments, at least in terms of the duration and the fluidity of the movement. I don't know if that was clear or not. But so discussion. So just I want to thank the Region Bourgogne-Franche-Comté for financing, and our colleagues at BioSerenity and colleagues at INSERM. And maybe some things that we could discuss are, in terms of rehabilitation, you could imagine if you had a patient in front of the robot. This is the kind of thing that Nicola does, I think, where you would dynamically monitor the behavior of the patient and modify the behavior you're asking him or her to make based on their behavior. So keeping kind of it in an optimal zone of learning. Then this notion of the EEG biomarker, issues of computational neurorehabilitation. We have some modeling work that I didn't feel like I had time to talk about for modeling this kind of behavior. So what I would hope we could do now is discuss your thoughts on how this might be useful, and if it rings any bells with things that you're doing or have thought about doing. So thanks for listening.
Thanks, Peter. That was really interesting. Early in my career, I did a lot of work with the serial reaction time paradigm. And I mainly used, instead of discrete movements, we did tracking tasks.
Okay.
And this was actually Laura Boyd's dissertation. And we looked at this with respect to where the lesions were in our individuals with stroke. And we used a similar analysis that you did. It was a cross correlation to look at the time lag, because it was very clear that as people were improving, with the repeated sequences, that they were clearly anticipating where the next, what the next element was going to be. And that was a source of their reduction in reaction time.
Yeah.
So we wanted to understand whether or not this capability was still there. And one of the interesting things that we found is that we had a group of patients with cerebellar stroke. So they had a lesion in the posterior circulation, just affecting the cerebellum, not the typical sensory motor area. And those patients could not acquire the temporal aspect of the movement. So they could not anticipate. Their accuracy was great, but their time lag never improved.
Okay.
Over practice. And so that was kind of that sort of told us something about at least the neural substrates for that aspect of this kind of learning. Okay.
And if you would give them a random probe sequence, would their reaction times go up?
Yes.
Okay. So they were learning. So they were learning the sequence, but but they couldn't anticipate.
Right. Exactly. And so we were able to dissociate that using that sort of cross correlation analysis.
Right, okay. Yeah.
where you sort of slide the, the spatial aspect until you get no time lag. And so until you get a high cross correlation
Right.
for the movement. And then you use the amount that you slid it to tell you what the lag was.
Right. Right. Yeah.
Is that the kind of analysis you're doing as well to?
Yeah, that I believe that's what the correlation function that we do kind of, it within a given limit that you can pre-specify of how far left and right will you let it look. And then it slides automatically and calculates where is the maximum correlation and the lag at that point.
Right. The other thing I wanted to mention is I think there's probably a rich amount of information in studying the actual movements, as you were discussing with Dr. Gordon. One of the things you might look at is dwell time on the actual, the point that they're supposed to go to. We found that particular measure to be very informative for when we were studying repetitive tapping movements. So how long do they spend, at that place before they initiate the next move? Of course, that may be dictated by following the robot.
Yeah.
I'm
But
not sure.
Yeah, but and that kind of comes back to what Jim Gordon was saying, that if you know the dwell time may also be affected by, well, I'm pretty sure where the next thing is, so I'm going to go ahead and get going even though I'm not positive. And so that there's some notion of certainty about the next movement that probably influences this dwell time.
Yeah. Great.
Okay, great. Yeah. Well, I'll look at those. I'll check out that bibliography that you're talking about.
Yeah, I can send you the papers.
Okay, that would be great. Kristen. Oh,
Hi, Nicholas was first. If you would like to go,
oh, sorry. Yeah, go ahead, Nicholas.
I have two comments and one question, the kind of devil's advocate question. So comments, I don't know if you know this paper that I published with Harris. Papagzantis, the last author on Peter's study, where we look at mental imagery on a sequence. And we measured, so it was kind of a complicated sequence similar to your sequences. Of course, we didn't make any changes to the sequence. That would have been interesting to do, but we haven't done that. And just by measuring the movement, so when you do mental imagery, you cannot do much. So we just measure the movement time of the sequence, of the imagined sequence or the real sequence. And we found very good correlate between the two. So it seems like this type of experiments have also been done in the past, but of course, in this case, there was no robot. It was just a little diode lighting up on the apparatus. So this is my first comment. The second comment is that in rehabilitation, people move away from robotics because the robots, they break, they're expensive, nobody knows how to maintain them. So people look for very simple devices. So that leads to my devil's advocate question is, why do you really need a robot? Can you have a little cursor moving on the screen instead, or showing a hand of somebody or something instead of the actual robot? So what is the robot buying you there? Mm-hmm.
Well, yeah, and so if you ask what can a robot buy you, and so like with respect to diodes or so what the robot can buy you is it can produce trajectories in three-dimensional space.
Mm-hmm.
And so if you think, so one thing that you could do is say, well, we want people to learn sequences in three-dimensional space. So there's a plane, but also depth. Mm-hmm. And then you could say, so I could be sort of my own devil's advocate and say, yeah, we're just going to invent a task that you can only do with a robot. And then we'll say, that's why we need a robot.
So someone called Jill Stewart, who was a student in our lab, and actually one of Caroli's students, but working with Jim Gordon, used VR to do this 3D movement as well.
Okay, yeah. There's also, and to be honest, like I like robots, and I have these robots. And when we came to Dijon, I was just thinking, we got to do something with the robot in the motion capture room. I mean, it's just too tempting. We have to figure out something that we can do with robots in the motion capture room. And so this was the first. Yeah. So it may be, that it could be that in the end you might say, we'll do fundamental research with the robot and then have kind of a patient protocol that could be deployed in homes or in different facilities that uses a screen, in fact, and the robot is just for fun for the researchers.
Thank you.
Hi, this was really interesting. Thank you. I have two comments. Well, and maybe, yeah, two comments. So one is about the measure or how to capture the variability. And I'm a gait person, so my awareness of the metric is in the gait world, but it's a measure that people use called the coefficient of correspondence. And it's looking at the difference between angle plots of the leg from one stride to the next. And so I imagine that could be extrapolated to a trajectory of the upper extremity as well, and comparing the robot versus the human. So that might be something to consider, but it doesn't capture time at all. It's just a spatial metric. And then my other comment, and this is not about the metric, which is what you asked for feedback about, but I'm immediately curious and I think there's a lot of maybe quite a bit of data about this in some capacity, where if people are practicing one serial and then introducing the perturbation, what the difference in learning is if you introduce a certain number of perturbations versus no perturbations, and if their overall learning by the last block is better if you increase the variability. Carolee has a paper that shows variable learning is better. And so I'm immediately thinking about that trajectory. And then they also used TMS to evaluate the neural substrates that are involved in variable task practice there. So I think it's an interesting extension of that work. Yeah, no, thanks. That's a good point. And in fact, just in some of the learning curves that we see, there's not. Yeah, you could argue that, oh, it looks like the perturbation, as soon as we go back to the original sequence, they're actually better than they were before the perturbation. So yeah, that's a good point to see if judicious use of perturbation can actually increase learning. Thank you. Jim? Thanks, yeah. Yeah, I just have two sort of comments. One is I've always wondered what real world analogs there are to a serial reaction time task. And I mean, it's a stimulus driven, it's an arbitrary sequence that's stimulus driven. And, and I'm not sure I know what is a real world analog in upper extremity, but in lower extremity tasks or in whole body tasks, the real world analog is learning how to dance. Yeah. Okay. So if you could get a robot that dances, you might have, an interesting paradigm. Because that's what, I mean, looking at your robot, that's what people do. They learn sequences, and they learn sequences by copying, typically with a rhythmic structure, that copying the movements of their partner or their instructor or whoever. So so I think it is very much a dance-like process. Yeah, or maybe I'm just trying to think of, that's an excellent comment. And I'm trying to think of like an upper body dance, and it kind of thinking of some Tai Chi movements are a little bit upper body. I mean, it's not full upper body, but it would be, we got to look and see, is there anything that people learn
Well, what was that dance that they did in the
Ma-
90s?
the
You know,
Macarena?
Macarena, you know. You know, But anyway, my other comment is that I think what would be interesting, and apropos of VR and virtual reality, what would be interesting here is to know what the eyes are doing, and whether eye movements are leading, you know, and how are eye movements corresponding to predicted robot movements? And you know, the reason I mentioned VR is that in some VR setups, you get the eye movements as part of the setup. So you know, that would be worth looking at if you had that kind of technology available.
Yeah, that's a good remark also. Yeah, indeed, you can probably, well, yeah, that would be a whole nother dimension, is to study kind of eye-hand coordination, and to see, you know, is the eye predicting where the hand is going to go and all this kind of stuff. Okay, yeah, no, that's a good point. Good points there. Yeah, and so keep an eye out for upper body dancing kind of protocols that exist in the real world. But I think yeah, going back to this stuff that, yeah, what you were talking about, these response specification paradigm, and looking at the, trying to look at the kind of some of the kinematic details of these movements, like the dwell time, but also,
well, you have the data. Basically,
Yeah.
the approach here is to take trajectories, and you've got like oodles of trajectories. You know, you've got hundreds of them, and you've got trajectories that are going in each direction. And so you can take those trajectories, and you can take the ones that correspond to predicted movements. And so you can say, this is what a predicted trajectory looks like. And I can send you some papers. We had, you know, approaches to using regression techniques and that sort of thing to say, okay, well, what you know, what does that trajectory look like on average? Okay,
Yeah.
Now you take trajectories that happen in the movements that are unpredictable or that change the spatial or temporal characteristics, and you can compare those trajectories and say these trajectories deviate in these ways from the trajectories. And it's a I mean, the advantage you have of having hundreds of trajectories is something that you can really exploit here. And they're all very highly specified, these trajectories, and highly overlearned. And I think that there's an opportunity to take and it can be automated, this analysis, in certain ways. So I think there's real possibilities there.
Okay, yeah, maybe we can talk about that. If you're interested, I'd be happy to talk with you more about that.
Sure, I'll send you some references, and then
Okay.
you know, sure.
Great.
Very good, Peter.
Yeah,
Thank you
because,
very
like,
much.
when you say,
It's 1 o'clock, so maybe we'll
Okay.
officially finish here. Thank you very much, and maybe a few of us can stay online here. Hope you see people
okay,
are
super.
living too.
Thanks, everybody.
I have to run myself.
Yeah, so